So let's begin and let's talk about the application of transmission electron microscopy in modern scientific research. The main moments that I would like to discuss with you today will be about electron microscopy basics, image formation and transmission electron microscopy, image acquisition intent, time application, limitations, and some information about processing of samples for the transmission electron microscopy. So what is this? First of all, we should understand that this is a scientific method which utilizes a beam of high energy electrons to study really small objects. And sure, we have some logical questions. Why we have to use an electron microscope? Why not, for example, light microscope? How small the objects are, how small the objects might be, and what is the minimal and maximal size of the objects. And in this case, we have to discuss some physical moments and basic of this method. So theoretically, maximum resolution that one can obtain with a light microscope has been limited by the wavelengths of the photons. So usually in the light microscopy, as you know, for illumination, we use photons, but the wavelengths of the photons, let's say, quite large. And as a result of this, resolution that can be obtained from the light microscopy less than we can use during the electron microscopy investigation, because for the electron microscopy research, in the method of transmission electron microscopy, we use electrons beam like a source of illumination of the specimen, and as a result of this, resolution of our method much greater than when we use light microscopy. Human eye is an apparatus that has finite resolution, and if you will calculate, we can understand that maximum resolution of our eye is just near the 100 micrometers. So, and our eyes work in the visible spectrum of range of light, and for this reason, certainly we have to use microscopes. Why we need to use microscope? If we want to investigate objects that's smaller than 100 micrometers, and the wavelengths of the light used in the optical microscopy, as I told, does not allow to resolve details smaller than half of its wavelength once again. And this limitation was described by Ernst Ebb in the last century, and for this reason, should to use a spectrum of electromagnetic spectrum of electrons that are much more this wavelength smaller than visible spectrum. That's why when we are talking about electron microscopy investigation, usually we can obtain only black and white image, not colorful, only black and white image, because we don't work in the visible spectrum of light. So for comparison, let's analyze optical phase contrast microscopy of the Escherichia coli. Here we can find some small objects, but when we use electron microscopy, we can find much more details. Like here, for example, even you can recognize inclusion bodies in the inside of these bacteria. That during the contrast microscopy, light microscopy, we can only suspect that we see something, but for more details, we need certainly another type of microscopy, electron microscopy. So a little bit history. So everything was started in 1897 when Thomson announced and the negatively charged particles, later termed electrons. After that, Broglie proposed that a moving electron has wave-like properties. So physical laws for these particles also worked and had the same formulas. And after that, Bush proved that it was possible to focus a beam of electrons with a cylindrical magnetic lens, laying the foundation of electron optics. And first microscope was invented in 1931 by Ruska and his colleague, and in 1939 Siemens produced the first commercial transmission electron microscope. And later in 1986, Ernst Ruska was awarded with Nobel Prize in Physics for his pioneering work in the development of the electron microscope. And later, not so far from today, in 2017, a group of researchers also were awarded with the Nobel Prize in Chemistry for developing of prior electron microscopy. And just for additional information, Ernst Ruska told that he was very much disappointed that now even at the electron microscope the resolution also was limited by the wavelength. So when he investigated this variant of microscopy, he was so excited that no limit of resolution. But after that, he was a bit disappointed because even electrons with their wavelengths has some limitation, but it was very powerful invention, and still we use electron microscopy for our research. So one of the possible classification of electron microscopy, we can group into big groups like fluid beam mode and scanning beam mode microscope. And transmission electron microscopy belong to the fluid beam mode. Also, we know about X-ray microanalysis, scanning reflection electron microscopy, and scanning transmission electron microscopy. So if you will compare optical microscope, TEM and SEM, what should we say? That for sure for TEM and for SEM, we use another principle, another variant of object specimen elimination. For the TEM and SEM, we use beam of electrons. It's very critical and important information. While during optical microscopy, we usually use light, and we can record it and we can analyze it with our eyes. I mean, information that we obtain from the optical microscopes. While transmission, particularly electron microscopy, require specific detectors like fluorescent screen, or it could be CCD camera for detection of the electrons that when pass through the specimen, electron beam bend in different direction. It depends on the density of the specimen structures, and as a result of this, we receive information on the fluorescent screen. So once again, if you will compare microscope, light microscope will show you much more detailed information and image, while light and scanning electron microscopy will give you much more information. So this is also comparison of TEM and SEM, just for your information, and you can find that you may recognize different details here. And scanning electron microscopy is a basic principle, and if you will compare with TEM, scanning electron microscopy give you information of reflection from the surface of the object. So transmission electron means that a beam of electrons pass through the specimen, while scanning electron microscopy show information when electrons reflect from the surface of the object. So that's why the image will be a bit different. But once again, only black and white imaging. So it's like a sort of limitation. If you see of this method, if you see colored picture, colored electronograms, that means it's just artificially colored, so in the Photoshop or in another software, but it never, just for illustration, just for simplification of analysis, but it's not a real coloring method. So what information can we obtain from the electron microscopy? Sample morphology. So when we are talking about TEM, we can recognize shape and size of the particles that you can find in the object. And when we are talking about the scanning electron microscopy, you can recognize sample topology, like a surface of the features of an object and its texture. Also, you can use electron microscopy for the sample element composition and sample crystallography. For example, how the atoms are arranged in the object, and etc. Main parts of the electron microscope. So the electronic part, and it consists of high voltage sources and sources for electromagnetic lenses, regulation circuits, and PC-based control. Vacuum part, because I didn't mention, but electrons pass through the electron microscope only in the vacuum. So, and vacuum part of the transmission electron microscopy usually realized due to different pumps. Conventionally, TEM has two pumps, like rotary pump for rough vacuum and diffusion pump for the more high and deep vacuum. So, and imaging part also is a part of electron microscope, and electron beam generation and manipulation may realize due to this, and image generation and magnification also. And image recording part, like digital recording through the CCD camera, or due to films. So we still use, some laboratories can use films for recordings of the electron microscopy images. So this is a modern electron microscope. How does it look in the laboratory? What can we find here? Electron gun, column, and here system of the electromagnetic lenses, like condenser and objective lens here, and see set of projector lenses that responsible for the magnification of our image, and here obligatory fluorescent screen plus image recording system also can be present in the electron microscope. So I want to explain you how can we receive this beam of electrons. From the top to down, electrons can be released from the cathode. Cathode can be different source. It can be from the tungsten or filament or lanthanum hexabarite, or some another single crystal source. And the main moment that this gun connected to the high voltage supply, and when we through the filament, for example, through the tungsten, pass current supply, this responsible for the emission of electron. We name it like thermionic electron emission. And as soon as these electrons released from the surface of the tungsten, high voltage source that connected to the gun of electrons, given sufficient current, and electrons can be focused and released in the column here. After this, due to condenser lenses, condenser aperture, these electrons concentrated on the surface of the object that we want to analyze. And after this, when this beam passes through the specimen, system of the projector's lens responsible for the magnification, final magnification of our image. So on this photo, you can find tungsten, conventional tungsten cathode. And this diagram responsible for the explanation of how heating of the tungsten cathode change the saturation of the beam, and we usually work with well-saturated source of electrons, because these variants are not suitable for the electron microscopy work. So usually when we adjust and prepare our electron microscope for the work, we saturated our cathode due to changing of the current that passed through the tungsten needle, and as a result we receive this well bright and well illuminated electron microscopy beam, and we're ready to start for our investigation. So as I told, there are different variants of the cathode materials, types. Usually in my lab I work with tungsten cathode because it's cheap, easy for replacement, and also operating vacuum is near this range, that easier to achieve. But also we can find and you can use different other variants. And as you can see, the cathode life approximately 200 hours. That also quite good. But different other variants of cathode can show even greater result. So on this slide, I just illustrate how can we change electron, how can we change cathode. So we just open column and lift cathode, lift electron gun, and here we can find this cathode and replace it. And after that, obligatory adjust, because the top of the needle of the cathode must be ideally in the center of the cathode gun, and for this reason we use different tools. And after that we again put this electron gun in the column and work with vacuum, pump vacuum, and adjust our beam to the working mode. So once again, the transmission electron microscope use fine electron beam created by voltage and electric current heated tungsten filament focused by magnetic lenses, and the electrons passes through the ultra thin plastic section and analyze that impregnated different heavy metals like osmium, like uranyl acetate, like lead, and after that the beam may bend in the different direction and in the different degree, and as a result of this, on the fluorescent electron we receive some image, and this fluorescent electron can capture this information, and we analyze in the binocular microscope all that we need. So this is fluorescent screen and image acquisition, yes, in TEM, and this is how does it appear directly when we work with the microscope. And this is a result of recording due to CCD camera. So once again, black and white. So this is green green color of the fluorescence is a result of illumination of this screen by electron beam. But also we can use for the acquisition not only direct screen, but we can use CCD camera and work with monitor, not with luminescent screen. As I told, the different laboratories still use films for the recording of the information, but certainly more convenient and more rapid variant is use CCD camera. So, TEM application. When can we use transmission electron microscopy? When can I advise to use it? Certainly, TEM still continues to play a critical role in this discovering and describing new organisms, especially viruses. And in this case, I can't ignore and obligatory have to say about our last big deal with coronavirus pandemic. So certainly, I'm sure that you saw these photos of the coronavirus viruses that appear like small round-shaped particles in the cytoplasm or on the surface of the respiratory epithelium, for example, like on this slide. And also, so new viruses and old viruses that are well known can be discovered and described due to transmission electron microscopy. Also, TEM has been critical in the HIV research, particularly in the area of the morphogenesis and the natural history of the virus and tissue changes in the disease and the identification and characteristic of the different opportunistic pathogens like adenovirus, cytomegalovirus, papavirus, microsporidia, herpes virus, and different other. So one moment. This slide also once again about viral identification in the biological samples, and here is comparison between SARS-CoV-2 and MERS-CoV, so they appear quite similar. And also TEM is very useful for understanding of the structure and organization of the components of tissue, cells, organism, how these organelles and compartments coordinate between each other. So certainly transmission electron microscopy is very important for the morphological analysis and widely used in this area. So on this slide, on this photo, you can find blood vessels in the right hippocampus, and you can recognize red blood cell, endothelial cells, and different tiny ultrastructures in the cytoplasm of these cells, in the endothelial cells. What else? Electron microscopic immunocytochemistry. It's like combination between immunohistochemistry and the transmission electron microscopy. It's on the cutting edge. So we can use different dense particles. Usually we use gold particles for highlighting of the different tiny ultrastructures in the cell. So how does it work? We use primary antibody against antigen and secondary antibody that coupled with a colloidal gold particle, and the size of this particle between 5 until 15 nanometers. And as a result of this, after a lot of preparation and sample processing, you can recognize different specific antigens in the cytoplasm of cell or on the surface of the cell. Like, for example, here and the big lymphoblastoid cell, we can find CD82 antigen domain in the MHC class 2 compartments. So that was investigation of MHC, main MHC complex. What else? Transmission electron microscopy is quite useful for the synaptic plasticity research. And here you can find combined immunoglobulin labeling for the glutamate receptors, and even you can use double immunoglobulin labeling. What does it mean? When you choose different size of particles for highlighting of different antigens. So only size or shape can be can be used for differentiation of different antigens. Because as I told, we use only black and white images, so we can't use different color of fluorophore like during fluorescent microscopy. We can only work with the black and white color, and as a result only with different sized particles or different shaped particles when we work with immunoglobulin labeling. Also crucial role of TEM in the autophagy research, and as you can see here autophagosomes that indicated by TEM, and also LC3 immunostaining. So this is from our lab, autophagosome in the hippocampus. And next variant of electron microscopy, also brand new, is correlative light and electron microscopy method that combines light microscopy with high resolution electron microscopy. And the combination of the various labels used in the light microscopy imaging with a nanoscale high resolution structure provided by M makes correlative microscopy superior for morphological studies of different types. No, in this case, once again, of autophagy. So what I mean that, for example, on this slide, these red arrows pointed different structure at the light microscopy level, and at the same time, also these 5 red arrows pointed to different structure at the level of the electron microscopy. And at the same time, you can use fluorochromes to highlight different antigens that in the area of your interest, and correlate these structures with electron microscopy on the ultrastructural level. So this is very powerful and useful tool for better analysis of the morphological structures. Next moment, cryo-electron microscopy. What is this? Cryo-electron microscopy, this method was awarded with Nobel Prize, as I told, in 2017, is a specialized form of electron microscopy where samples are frozen in non-crystalline form of ice, that termed vitreous ice, which allows images of sample captures in their native state, and this native state can be visualized. Cryo-M can be applied to purified proteins, for example, that are frozen in very thin vitreous ice, so that only a single protein is present, for example, in any section of the ice, generating an atomic level 2D picture of that protein. And however, during freezing, that proteins will have multiple different orientations, with each of these leading to images of the protein at the different angles. So as a result of this, software can combine all these images and perform 3D reconstruction of the protein that in area of our interest. So on this slide, you can find a lot of pictures, a lot of images that captured during the examination, and after that, taking into account that all this protein were fixed in the different direction, software can build up 3D reconstruction of this protein. What I have to say that for this method, we have to use cryo camera for the analysis, and also our specimen must be freeze in the nitro oxygen. So this is the another variant of the cryo images, and as you can maybe recognize, this is structure of the spike protein of the coronavirus, and this is SARS-CoV-2 virus reconstruction due to cryo. I guess that all of you saw these slides, because coronavirus pandemic was very powerful experience for all us. And everyone was interested in the brand new researches in this area. So what about the application of the TEM in pathology? First of all, TEM still a very useful tool for the diagnosis of the renal biopsies. Also, we use TEM for the ciliary pathology. I mean, when we investigate ciliary epithelium of the respiratory tract. Storage diseases also require TEM examination, neuromuscular diseases, and even when we are not sure about source of metastasis for the tumors with unknown primary localization, we also can use transmission electron microscopy. And in cytopathology, transmission electron microscopy is also very useful. This slide is about the contribution of this method for the evaluation of native kidney biopsies. And as you can see, that a lot of diseases of kidney apparatus still require electron microscopy examination. So this column indicates that this method is essential for diagnosis. And this column is about helpful and not required. What about the limitations of them? Certainly this method has his advantages and disadvantages. And first of all, these methods are large and very expensive. And another moment is that this method require laborious sample preparation. So it's a huge of work to prepare really well specimen for the analysis, because as I described all these steps and structure of the electron microscope, and for your understanding that it's high-tech method, and for sure for this method we need very perfectly prepared specimen, tiny, thin, and well contrasted, and this requires a lot of work. And I will give you some highlights about the stages of sample preparation. What else? Potential artifacts from sample preparation. So we use fixation and let's say dead material, so it's not in vivo examination, and certainly it leads to the different artificial changes of sample. So operation and analysis requires special training. Yes, sure. And it's not so easy to work with this device, with this microscope. Samples are limited to those that are electron transparent and able to tolerate the vacuum chamber and small enough to fit in the chamber. What that does it mean? That means that for electron microscopy, we usually use very small pieces of tissue, only 1 millimeter in square volume, because we can't put large-sized sample in the vacuum chamber for analysis. So usually we use really small pieces of tissue, but at the same time we can obtain a lot of information from this small volume material. And certainly this section of the sample must be very thin, because through the thick section, electron beam just can't pass through. That's why we will use not just conventional microtom for paraffin section, we will use ultra microtom. I also will show you how does it work. And TEM requires special housing and maintenance. Yeah, that's true. Microscope very very complicated, and at the same time require a lot of attention, because of when I talked with another electron microscopist, all of them told that every week something happens with this device that require new and that needs some specific maintenance and adjustment. So you have to work and control this device, this microscope, every time when you prepare for the work. And TEM routinely provides back to wide scale and two-dimensional image. Yes, that's true. And what about the main stages and timing of processing sample for the TEM? So in my lab, usually this needs 4 or 5 days for preparation, because we have different stages, different steps for sample processing, from primary fixation in the glutaraldehyde. So we don't use formaldehyde for this reason. We can use, but some in solution with glutaraldehyde usually. And also after that we use secondary fixation with osmium tetroxide. Why osmium? Because it's like additional contrast of our specimen. After that, dehydration with uranyl acetate, once again heavy metals, and it had to be have to be impregnate different structure of our specimen. Infiltration of resin, it can be RLD, it can be EPON, or it can be, for example, white. So different variants of resin may be used. I usually use EPON RLD mixture. And after that, curing of our blocks in EPON, for example, and blocks section. So this part, I mean, section very very laborious also moment, because you can be very attentive and exact during this stage. So these slides describe how different concentration of glutaraldehyde can influence on the ultrastructure, in this case in the red kidney. So between 1 to 4%, we usually use 2% glutaraldehyde. And what about the choice of fixative agents? So as I told, we have primary and secondary fixation, like osmium tetroxide and glutaraldehyde, but usually we use combination of these fixative liquids. And factors that affect the quality of fixation, it's concentrational and length of fixation, temperature of fixation, pH, and osmolarity of fixation solution, purity of aldehyde fixation, tissue type, method, and rate of fixation penetration. So this is a different time and temperature of fixation. So usually we recommend hours. But even after the years, when you just, for example, forgot your specimen in the freeze, you still can receive some information even after 11 years, for example, like in this case of the fixation. So we obtain our blocks in a phone. This how does this block appear in the molds, and after that you will use a knife for the cutting of this block, for the trimming and cutting. So usually in my first years I used a glass knife from the glass, but after I was level up and used diamond knife. Certainly, what is the difference between glass knife and diamond knife for the cutting of the ultra-thin section? It's the duration of the usage. How long can you use this knife? So glass knife only for let's say 20 sections, while diamond knife you can use during long time, years, for example. And this slide illustrates semi-thin preparation. This is under the microscope. So when you cut this slide, the block, you obligatorily look in the binocular, because this very tiny block, and this is how does it appear under the microscope. This is semi-thin section. And after that, when we receive this semi-thin section, we put this semi-thin section on the slide and can stain with or toluidine blue or methylene blue, some stain that can penetrate in the resin, and estimate first impression of our block we receive from the light microscopy. And when we realize which part of the tissue we would like to analyze under the electron microscope, we just cut in the block again, and with the area that an area of our interest. So we cut once again this block, choose specific place for the further investigation, and once again cut. But now we cut at the another level of the section thickness. I didn't say that, just for comparison. Light microscopy use section with thickness 3-4 micrometers. I mean paraffin embedding specimens, 3-4 micrometers. Semi-thin section, usually 1 micrometer with thickness. Ultra-thin section, and now look, this is a color scale of section thickness that we can estimate with just reflection from the surface of this thin section. We use 70-80 nanometers of thickness of our section. So gold standard for analysis of section is silver section, and on this slide and on this photo you can find the ribbon of the section with silver color of the surface. So this means that this is approximately 70-80 nanometers of thickness on this slide of this section. But depends on the aim. For example, for the ciliary pathology, when we analyze ciliated epithelium of the respiratory tract, we need thick electron, thick ultra-thin section. Thick means 90 nanometers. And for example, electron microscopy tomography, when the aim is to receive 3D model of the ultrastructure, we can use even thicker sections like 200 nanometers, for example. So this is how does it appear. So we have a foam block, resin block, and here black colored particle is our sample. Why black? Because during processing we use osmium tetroxide, and as a result of this, a lot of heavy metals impregnate different structures of our sample. And after that, we can cut it and receive this perfect ribbon of the sections that after will be placed on the supporting grid or supporting mesh. This grid can be from different metals. It can be copper or nickel or silver grids or even gold grids, so depends on your aim. So the cheap and commonly used is copper grids, certainly, but when you need to work with immunogold labeling, you can't use copper grids due to chemism of chemical specialties of the process. You need to use nickel grids like here. So grids, holders, and anti-mag tweezers, our stuff for the collection of the sections on the surface of this grid. It's really very, very tiny structures. So after that, we have to enhance our contrast, and for this reason, we use uranyl acetate and lead citrate. Next step, we put our grid in the holder that finally we will put inside the column, and after we receive this picture on the surface of our fluorescent screen. This slide shows that we can even reconstruct some organelles or structures due to serial ultrasin sections, and this was also made in our laboratory, like in this case mitochondria modeling. Immunogold labeling, once again, it's also we perform in our laboratory. And for this reason, we use different steps like Bowen serum blocking after the primary antibody, secondary antibody application, and after we, for example, use synaptophysin for the hippocampus of the rat, and receive this slide that illustrates the accumulation of the antigens in the ultra structure of the hippocampus of the rat. Not only for the tissue we can use this method, but also for the suspension of the cells, for example, for the platelets, like on this slide, or even for the organelles. And this slide illustrates mitochondrial suspension in the CA1 region of the rat hippocampus. So this was my last slide, and thank you very much for your attention, and I will be glad to answer any question. Thank you.
Thank you very much for this wonderful detailed explanation of electron microscopy. Thank you. It was really interesting. Well, dear participants, do you have any questions? You can write them in the chat and you can ask them directly. So while our participants are getting ready, I have a question. As far as I understood, Well, first, my first question was how much time does it take to prepare the samples? And I understood that it takes 4 to 5 days.
Yes, it's not so quickly.
Is the procedure the same for the viruses, bacteria, and tissues, or it is different?
No, you can. No, so certainly you will use some special conditions for the viral preparation, safety conditions certainly, and for viruses, taking into account that it's a smaller structures, you need less time. But from my experience, we use with biological samples, not with the viral, because and microbes, because we was not certified to work with this type of material.
Mm-hmm. And what about the samples with coronavirus? These were your samples?
No.
Oh, okay.
We're not certified. In Ukraine, we work only with just biological samples from the tissues, biopsies from our patients. But
okay.
we can use we can analyze, for example, coronaviruses in the respiratory epithelium, and but we didn't do that during the pandemic.
Okay, thank you. So we have some questions in our chat. From Inna Junko, can electron microscopy be harmful to health of the specialist who does it?
Yes, and I hid this slide. One moment, I know how can I come back? One moment. Yes, sure, it can be very harmful, and for example, as a result of usage of the tetraoxide of osmium, or for example, propylene oxide, also very harmful and carcinogenic. And one moment, I want to show you this slide. I didn't, if I have time, certainly. Okay, it's not time to find it. Yes, electron microscopy, very harmful methods. So we have to follow all safety's rules, because it's tetraoxide osmium, very harmful for our air tract, propylene oxide and resins harmful and toxic and can be carcinogenic. But usually you work in the hood and in the gloves, in the white coat, and it can prevent all side effects of this method. So,
okay, thank you. We have a question from Mikhaila Ivansov. How do you think what methods of investigation will come after electron microscopic techniques?
Come after? No, already came. It's cryo-electron microscopy, when we can analyze in an atomic level of the structures, and the resolution of the cryo-electron microscopy really impressed. And nowadays, this method can be very useful also for the analysis of the stem cells and stem cells differentiation, on which in which line semi-pluripotent cells, let's say, will differentiate it, in which direction.
Thank you. Dmitry Alexandrovich wants to know how long you work with electron microscopy, and do you get pleasure while you're working?
Thank you for this question. I started my work with electron microscope on the 5th course when I was a student. It was in 2009, so 13 years ago. And it's not easy, because if to say truth, this tool very complicated, and all the time something is broken. All the time you have to fix it. And But I really love when I work directly with this tiny specimen, when I work with electron microscope, and when my job, I mean sample preparation, was done at the excellent level, and I see all details. That's really, I really enjoy it. When I can estimate these tiny structures, it's like, you know, space investigation, but at the nano level. So yes, I really love it, but it's not easy, but it's fun, sometimes really fun. Okay.
It's great, and our participants wish you to take care of yourself also.
Thank you very much. Thank you very much.
And Olga Gavriliev has a question. What is the power of electron microscopes in your laboratory?
100,000 maximum, but for the biological specimen, for the tissues, we usually use till 20,000. So it's from 2,000-3,000 of magnification till 20,000. So it's our common range of magnifications. My PhD thesis was about intercellular junctions of the heart, and I used 80,000 magnification for the gap junction analysis, but it's rarely, really rarely we use such high magnification. For biological samples, we use, as I mentioned, 3, 5, 10,000 of magnification.
Thank you. Oh, one more question.
Mm-hmm.
How long did you practice electron microscopy techniques to use them fluently?
3-5 years, maybe. It's not quickly, yes, because sample preparation and operation with STEM, it's not so simple technique. So you need to be experienced. That's why electron microscopists, specialists, not so many, and we also know each other and all the time support each other, because it's hard way, but we still on them.
Are there any general trainings, or it depends on the microscope that you are going to work with for microscopists?
Once again, because it was interaction.
Are there any trainings for electron microscopists, or does it depend on the exact microscope you are going to work with?
No, that's general course of electron microscopy, and my first site on this method I received in the Ternopil with Professor Volkov, and it was 5 days of training. 5 days of training, and after that I investigated by myself. But sample preparation is the standard with different maybe specific details for different tissues, as you asked about, for example, viruses or maybe cell culture. But if we are talking about electron microscopy operation, certainly it depends on the type of the manufacturer of this, but general rules are working. So,
Thank you. Thank you very much. Thank you for your question, for your answers.
Thank you.
Dear participants, any more questions? Well, everybody's thanking you on the chat.
Thank you very much for attention.